So we are still in the process of trying to sequence a genome. We have purified our DNA from our cells or organism of interest. We have fragmented that DNA into more usable fragments, and we have now amplified those fragments using polymerase chain reaction. Now we need to actually sequence these fragments by assigning nucleotide bases to the fragments themselves. So there are different types of sequencing that we can do. The cheapest and the easiest one is called Sanger sequencing. It's also known as the dideoxy method, and that is because it uses dideoxynucleotides that have a hydrogen instead of a hydroxyl group on the 3' carbon of the sugar phosphate backbone. How it works is the amplified fragments are going to be replicated all over again, but now instead of in the presence of just normal nucleotides, it will be in the presence of normal nucleotides and dideoxynucleotides, again with that hydrogen on the 3' carbon. The significance of this hydrogen on the 3' carbon is that it will stop replication from happening because there is no available 3' hydroxyl group to make a phosphodiester bond. So replication enzymes will use normal nucleotide bases and then will randomly insert these dideoxynucleotides that will stop replication. These dideoxynucleotides are going to be tagged to fluorescent proteins or are going to be tagged to different radioactive isotopes, and they're going to be specific for each nucleotide. So we're going to have one type of fluorescent tag that is going to be assigned to C bases, one to T, one to A, and one to G. And then after we are done this replication or this amplification of our DNA sequences, we can run our fragments on a gel. And we can subject that gel to a fluorescent filter. And then the order of the sequences from shortest to longest, as the fragment that traveled the most or traveled furthest down the gel will be the shortest, and the one that traveled last up the gel will be the longest, will tell us what our sequence is. And this is most useful in sequencing when you want to sequence single genes rather than an entire genome at once. So this is an example of what Sanger sequencing looks like. So here you can see that we have black assigned to G, blue to C, green to A, and red to T. We'll use our normal polymerase chain reaction idea, but instead of using just normal nucleotides, we will also incubate with our fluorescently tagged dideoxynucleotides, which are signified by this little hydrogen group here. As you can see, the polymerase will integrate a dideoxynucleotide randomly while it is replicating, which will stop replication. Then you can run these fragments on a gel, which will then subject it to a fluorescent filter, which will tell you that it goes from A, C, T, G, C, T, T, etc. And then what Sanger sequencing usually gives rise to is a chromatogram, which will actually tell you the peaks of the fluorescent signal that is being given off to tell you what the sequence is. Another type of sequencing is called whole genome shotgun sequencing. And unlike Sanger sequencing, that is really powerful for sequencing a single gene, it is much more useful for sequencing an entire genome. So how it works is you will isolate genomic DNA, and again, similar to how you would do it for Sanger sequencing, you would have to break it up into overlapping fragments. And you would do this by using two different DNA samples that have been digested by two different restriction enzymes. And this is how you're going to have those overlapping fragments. You're going to clone each fragment into a plasmid expression vector, and then you can sequence the genomic DNA fragment in each clone. So again, we're still going to be going kind of back to using Sanger sequencing, but now we're only going to be sequencing smaller DNA from each of our bacterial colonies. Then, why this is faster than just using normal Sanger sequencing for an entire genome, you will use a computer program to realign your overlapping sequences. The most powerful, but also the most expensive tool for sequencing is called next-generation sequencing, and it's useful for when you want to sequence multiple genes or a whole genome. How it works is you need to ligate adapter sequences onto the ends of all of your amplified DNA fragments. So you would have gone through isolation, you would have fragmented, you would have amplified, and then you would have put adapter sequences onto the ends. These adapter sequences are going to allow your fragmented DNA to be immobilized, or like literally to stick onto a DNA array. This DNA array is then going to allow a detector to measure or capture a fluorescently labeled nucleotide as it's being amplified. So the computer, it will look at a single spot on this DNA array, and then as a like red, green, blue, black, as that's being shown, it will tell the detector exactly what nucleotide is being put into that specific location. And so in comparison to the other previously mentioned sequencing techniques, it's almost instantaneous for you to actually get your sequence of interest, but also comes at a higher price tag. Given the following Sanger sequencing results, which of the following is the correct sequence of the template DNA? So before we even look at our answer choices, let's just figure this out based off of our Sanger sequencing results. So we remember during Sanger sequencing, we are amplifying a strand of DNA using dideoxynucleotides, where when we have this dideoxynucleotide, it has a 3' hydrogen instead of a 3' hydroxyl group. And this will cause replication to stop. And then based off of the fragment length, we can then know where this nucleotide has been incorporated into the nucleotide chain. So if we're looking at our Sanger sequencing results, we know that the further down the gel that a nucleotide sequence has run means the smaller it is. So the closer to the 5' end that it is. The further up the gel it is, the longer it is, meaning to the closest to the 3' end it is. And why do I know this 5' versus 3'? Well, in order to actually grow this nucleotide chain, we have to add nucleotides onto the 3' hydroxyl group. So that means that during Sanger sequencing, the polymerase that is actually doing this is going from 5' to 3'. So let's figure out what the 5' to 3' DNA looks like based off of our results. So we're going to go from bottom to top, where we know that 5', the smallest one, is going to be a T. Then we're going to have an A. Then we're going to have 2 G's next to C, then 2 T's next to C, then G, T, A. So we're not done yet. Because the question is looking for the template DNA, this is the sequence of our complementary or the actual Sanger sequence strand. But we need to know the 3' to 5' strand. So now we're just going to do our complementary base pairing. And now we can actually look at our answer choices. So the convention when it comes to listing DNA sequences is to go from 5' to 3'. So if we're looking at our answer choices, generally speaking, we're going to want to convert our 3' to 5' strand into a 5' to 3' strand, where literally we're just going to copy the strand, but just in the opposite direction. So if we look at our answer choices, the one that is in the 5' to 3' orientation is going to be D, because you can see we have TAC, GAA, GCC, TA. Technically, answer choice C is also giving us the exact same sequence, just in the opposite direction, because this one would be the 3' to 5'. However, because the question isn't explicitly stating that is looking for the 3' to 5' sequence, then you have to assume that it's giving it to you in the 5' to 3' sequence, leaving us with answer choice D.